This is audio assignment 3-1, Introduction to Hemostasis. Hemostasis, which is also known as the hemostatic process, is a vital function of the human body and is a clotting response that literally means to stop the flow of blood. The hemostatic process is a complex, coordinated interaction between our endothelial cells, and our endothelial cells line vessel walls. It also involves platelets, coagulation or clotting factors, and other types of plasma proteins. And I mentioned clotting factors or your coagulation factors. And these are the coag factors are labeled using Roman numerals. And if you're not familiar with Roman numbers or Roman numerals, be familiar with numbers 1 through 13 so you can recognize those if you see them. Doctors can order tests on individual clotting factors, so you do have to be able to know what the Roman numerals in reference to. You're going to also notice in upcoming slides that the Roman numerals used are not in order or in sequence of 1, 2, and so on, but they are arranged in order of discovery of that particular clotting factor. So during the hemostatic process, each individual factor is going to activate the next in a sequence, one right after the other, and we refer to this as the coagulation cascade. There are two pathways for clot formation. There's primary hemostasis, which involves vasoconstriction and platelet plug formation, and secondary hemostasis, where we have the final fibrin clot that forms. And we're going to start off by talking about primary hemostasis, vasoconstriction, and platelet plug formation. This slide you're going to see is the process of hemostasis. It's a good explanation of the physiology of blood clotting. So you notice that here is our sequence: vasoconstriction, platelet plug formation, then we have our stable fibrin clot that forms, and at the end is fibrinolysis. We're going to go through each of these steps individually. Okay, so in this slide we're going to look at vasoconstriction. First step in the process. And if you look at the term vaso, that means vessel, and constrict means to tighten up, so that's exactly what's going to happen here. So you have an injury to your vessel, notice you're bleeding out, and this injury is going to trigger the release of tissue factor 3, and this is also known as tissue thromboplast, and this is going to initiate the clotting process. It's going to cause the injured vessel to start to narrow or constrict, and this is done with the help of smooth muscles in your vessels. It's to slow the flow of blood or slow the bleeding. And we also have collagen that's present in the connective tissue of the skin, and that when that becomes exposed, it provides a surface for your cells to attach to. Um, in a healthy individual, you have undamaged tissues, and there's collagen in the tissue, and it helps support blood vessel wall and the surrounding tissue, but it's concealed or it's covered by that endothelial layer of cells, and you can't come in contact with any circulating platelets. However, should that endothelial cell layer be removed either in disease or on tissue injury, then you're going to have the collagens revealed, and they're going to interact with your cellular components of blood as well as the proteins in the blood plasma to start to close and seal off that opening. Okay, so this is a figure diagram. It comes from your textbook, and it illustrates a megakaryocyte in box A. It's releasing a platelet, and the megakaryocyte is the name of the cell when it's found in the bone marrow. There are no platelets actually in the bone marrow. In the bone marrow, it's a megakaryocyte. You'll notice illustration B, that diagram shows the internal structure of a platelet. Platelets don't have a nucleus, but it does have other genetic material included, like mitochondria there. You see that's going to help with energy, things like that. Um, so, as I said, you can see the internal structure of the platelet. and platelets get released from the megakaryocyte. They're going to enter the circulating blood or they're going to be in the spleen. And I just want to mention here that platelets actually have the ability to release over 40 substances from its structure, so their involvement in clotting is really high. Okay, so we're still talking about primary hemostasis. We've got vasoconstrictions already happening. Now we're going to the second step. It's called platelet plug formation. And under the category of platelet plug formation, we have two basic steps, and it's called platelet adhesion and platelet aggregation. And the slide you're looking at is a diagram of that process. I'm going to explain that to you. Okay, so during platelet adhesion, you have damage that's going to expose the platelets to a clotting factor. It's called von Willebrand factor. You'll see that listed in the table that I'd mentioned earlier as von Willebrand factor. And von Willebrand factor is a glue-like substance that actually causes platelets to stick to damaged tissue. So it's going to work with that and the collagen in the endothelium to help the platelets start to stick. At this point, there is a small amount of thrombin created, and platelets are going to start to degranulate or flatten out as it's exposed to that thrombin, and this is where the extrinsic system is activated. We're going to talk about the extrinsic system here in just a little bit. So once this all started, other clotting factors are also going to get involved, starting to produce more and more thrombin. And the role of thrombin is to bind platelets. Platelets are going to start to change shape, going to completely degranulate, flatten out, start to develop spikes or spicules on their edges, and they're going to stick to one another for the next process called platelet aggregation. So in platelet adhesion, platelets are sticking to the vessel wall, sticking to each other. In platelet aggregation, we have tissue factor that's produced by the injury, as I mentioned. It's going to cause the platelets to move to the site of injury. It recruits other platelets to come to that injured site and to stick to one another, and this is where we have a platelet plug. That term aggregation means to move to or hang out, so platelets are moving to that injured site to hang out together, start to stick to one another. Now, all this can only happen if there is a normal concentration of platelets in the blood, or otherwise known as a normal platelet count. And I'll say that platelets should range between 140 to 440,000 to 450,000 for to be normal and all this to function correctly. All right, so now we're moving into secondary hemostasis. And like I mentioned before on table 3-1, you have a list of plasma proteins. These are your clotting factors that are found in blood, and all of them are proteins with the exception of your ionized calcium. Um, you're going to note, for example, on your table that there is a factor 8, and factor 8 is anti-hemophiliac factor. And that term anti, it means against or against something. So anti-hemophiliac means that it prevents hemophilia. Now, hemophilia is the name of a bleeding disorder, and it causes abnormal bleeding or bleeding that just won't stop. So people that have hemophilia have too little or even no factor 8 uh, in their blood. Some of your clotting factors also are vitamin K dependent. You'll see that in your reading, so you have to have a good diet with vitamin K in it. And you're also going to notice in your coag cascade that we also need calcium because that's involved in the clotting process. So calcium in the diet is also essential for good clotting. So at this point in the cascade or in the clotting process, this is where secondary hemostasis is coming into play. Secondary hemostasis involves the formation of a stable blood clot, and usually secondary hemostasis is needed for more serious injuries. If you have a venipuncture, the first phase is primary hemostasis. The first phase of um, which would be vasoconstriction and platelet plug formation. That functioning properly is all you need for a venipuncture. But when you have deeper injuries, you're going to need more or better or a more intense healing process. So, during secondary hemostasis, it's going to involve the amplification, which means you're going to intensify something, and the propagation, which means that it's proliferating or making more. So, we're going to have large amounts of thrombin needed to make strands. Fibrin strands. As more platelets are drawn to the injured site, you get even more burst of thrombin. That's going to trigger the activation of clotting factors on the surface of platelets and then causes fibrin strands to cross link into a stable mesh. And the mesh forms a net like structure. And this is going to trap blood cells and platelets to form a stable clot. For the most part, your coag factors and their interactions together can be summarized into actually two reactions: the conversion of prothrombin to thrombin, and then the conversion of fibrinogen to fibrin. Okay, so here in this slide, you see our coagulation pathways. This is how we've gotten from primary hemostasis down to secondary hemostasis. I referred to this coag cascade in the beginning of the slide. Um, the pathways are divided into three: your intrinsic pathway, the extrinsic pathway, and then the final common pathway to get to your stable fibrin clot. Notice in the extrinsic pathway, the little green box, you'll see that you have tissue thromboplastin, as I mentioned before, that initiates or it's released from an injured site. It's tissue factor or factor 3 there. And notice that that involves calcium. Ionized calcium here in the process. Also notice that for your Roman numeral 7 VII, notice there is a subscript letter A next to it. That means that it's been activated. So factor 3 triggers factor 7, and then it becomes activated. So as I said, this is a simplified version of the coag cascade. So extrinsic, let's talk about that. It means originating outside, okay, because it's activated by proteins outside of the vascular system or outside of your blood vessels. So you can think of it as it's been activated by external injury. Okay, it's pretty short, this process. You can see there's only a few things that it has to get through before it reaches the common pathway. Lasts about 30 seconds, and it takes about that long, 30 seconds to a minute, for you to stop bleeding from a venipuncture site. So really the extrinsic pathway is what's involved in the healing portion of just a plain old venipuncture. External injury, right? I just want to say too here that the extrinsic system is evaluated uh, in the lab using the coag test called the prothrombin time, pro time, or the PT. So that's three ways of saying PT. Then we have the intrinsic system, and that means originating from within, because it involves coag factors circulating within the bloodstream, and it is originated in when you have a deeper injury, internal types of injury. And you can see there are more clotting factors involved here. It is longer, the process is longer because of those, and it takes about 3 to 5 minutes for the cycle to complete. Your intrinsic path is evaluated using the coagulation test APTT, activated partial thromboplastin time. Now, most often the A is not mentioned in the test. We just call it the PTT for ease of conversation. We docs order PTTs a lot together, so that's going to be the reason that was in conversation that we're going to drop off that A. So at this point, both paths are going to join to form a common pathway, and it's going to form a solid fibrin clot. So, how does this clot dissolve? Okay, this is the process called fibrinolysis. You have your fibrin clot, and then it needs to be broken down or lysed. Um, Plasminogen is involved in this. Uh, it activates plasmin, and plasmin is the principle of our clot lysing system. It has a high specificity for fibrin and a particular ability to dissolve our fibrin clots. Once that clot dissolves, the resulting products are going to be excuse me, fibrin degradation products, the fragments of fibrinogen, or it can be XDPs and D-dimers that are cross-linked degradation products. We do have a couple of mechanisms that control our ability to clot, our normal fibrinolytic mechanisms that we've just talked about, along with other things that regulate and inhibit systems. This all prevents an isolated injury from initiating the clotting mechanisms. You would hate to have just some small injury and then all the clotting mechanisms start to come into play. This causes certain bleeding disorders. Your endothelium lining of blood vessels have been mentioned. Those are types of control mechanisms protect collagen from getting released, exposed to platelets. You have certain plasma proteins, and these all prevent unwanted clots. All these proteins are labeled C, S, and Z, or we have antithrombin 3, and that's just to name a few of the fibrinolytic mechanisms. We also want to mention to you that the liver is involved in hemostasis. It produces most of our clotting factors, and as I said before, we need vitamin K, and the bile salts the liver produces helps with the absorption of vitamin K. So if you have some sort of liver damage or something like that, you're going to have increased coagulation problems, specifically with the prothrombin. A couple of terms you should be familiar with, thrombus and embolus. The term thrombus means clot, and then the term embolism or embolus means obstruction. So you can put those two terms together, thromboembolism, and that means that there are blood clots caused by damaged vessels, or it could be heart valves, or it could be overactive coagulation that can result in a clot being transported actually to another location in the body, which can cause obstruction where it's formed. We'll talk about bleeding disorders later on in this section, or actually you'll read about them. But most coagulation problems that occur have to do with the formation of clots. So there's quite a few patients on types of anticoagulant therapy, and I've got a few drugs listed here. The first one, warfarin and Coumadin, is fairly commonly used, and warfarin, Coumadin, as patients show up for blood draws for this, we're going to draw the PT. That is what the PT, or the ProTime, is what's used to evaluate this type of anticoagulant therapy. And then patients taking heparin are going to have the PTT drawn from them. And for patients using Plavix or just taking aspirin as a blood thinner, aspirin and Plavix are antiplatelet medications, and there's platelet factor assays out there now, such as Verify Now. There's a P2Y12 system that measures platelet responses. I will say, though, that Plavix does have a black box warning that mentions that genotyping is the testing that should be done to check the effectiveness of Plavix. However, that's fairly expensive to do, and it's also laborious for laboratories. Most of them would probably have to send it to a reference laboratory. So many of our laboratories have decided to use platelet factor assays to measure response to Plavix or aspirin therapy.